Today, we're going to solve a mystery. We're diving deep into a new study that completely flips our understanding of how our brains age. We're going to follow the clues of a fascinating cellular investigation, one that challenges what we thought we knew about why our brain cells decline, and, get this, it offers a real hint that the whole process might actually be reversible.
Seriously, just think about that for a second. What if all the wear and tear, the slowdown that comes with decades of life, could just be undone? That's the huge, mind-blowing question at the very heart of this research. But to even begin to answer it, scientists had to crack an even bigger problem. How in the world do you study an 80-year-old brain cell without, waiting 80 years?
Well, the answer, as it turns out, is pretty incredible. You build one from scratch. Our investigation starts right here, with the amazing experimental setup that made this whole thing possible, figuring out how to literally create an old neuron in a lab dish.
So, how did they pull this off? It's a really slick four-step process. First, they took simple skin cells from people of all different ages. Now, these cells, called fibroblasts, have this amazing ability. They actually remember how old they are, biologically speaking. Then, with a special cocktail of molecules, the scientists didn't reverse the cell's age, they just gave it a new career path, switching it from a skin cell to a brain cell. And in just 30 days, boom, they had living, firing neurons that were, for all intents and purposes, 84 years old, right there under a microscope.
But of course, they had to be absolutely sure these were the real deal. So to double-check, they used these special fluorescent tags. These tags were designed to only light up and glow red once a cell had fully become a neuron. This was awesome because it let the researchers literally watch the transformation happen in real time. With their aged neurons now ready to go, the real detective work could finally begin.
The very first clue in our cellular mystery brings us to the mitochondria. You've probably heard of them. They're the tiny power plants inside every one of our cells, churning out energy. For a long time, the main theory about aging was that these power plants just get old, they break down, and they fail. So naturally, that's the first place these scientists looked.
And to check on the health of these power plants, they used this clever little tool called a JC1 sensor. It's basically a dye that changes color depending on how well a mitochondrion is doing its job. If the power plant is running great, putting out lots of energy, it glows a healthy red. But if it's struggling and its energy potential drops, it glows an unhealthy, sickly green.
And when they looked at the results, well, it was pretty obvious. The neurons from older donors were just full of these unhealthy, green, glowing mitochondria. So at first glance, it looked like the old theory was right on the money. The power plants in aging brain cells were failing. Case closed, right? Wrong. Because this is where the plot twist comes in. When the researchers looked closer, they found something that made no sense at all. These struggling, unhealthy power plants in the old neurons were incredibly busy. They were way more active than their healthy younger counterparts. Now, why on earth would a failing power plant be working overtime?
To understand what was going on, we need to know a little bit about how mitochondria behave. See, they're not just static little beans. They're constantly moving, splitting in two, that's called fission, or joining together, which is called fusion. It's how they manage quality control and share resources to stay healthy.
And this is the bombshell. The neurons from the older donors had a massive increase in both splitting and merging. The mitochondria were in a state of frantic, hyperactive chaos. This wasn't a sign of just giving up and failing. This was a sign of pure desperation. They were trying to salvage parts, to dilute damage, to do anything to survive. The power plants weren't just dying, they were fighting for their lives. So the real question became, what was putting them under all this stress?
To answer that question, our investigation moves from the power plants to the cell's sanitation department. It's a process called autophagy, and it's basically how a cell cleans up after itself, getting rid of all the damaged parts and garbage. The scientists started to suspect that the problem wasn't with the power plants at all, but with the total failure of the cleanup crew.
So they used another cool fluorescent tool to track the cleanup process. Let me break it down. The cell puts its trash into these special bags that start off glowing yellow. Now, to actually recycle that trash, the yellow bag has to merge with the cell's recycling center, which is full of acid. When that happens, the acid changes the bag's color to red. So yellow means trash has been bagged up, but red means it's actually been taken out and processed.
And the result here was absolutely shocking. In the young, healthy neurons, about half of the trash bags turned red. Pretty efficient, right? But in the neurons from older donors, that number just tanked, only 16.6%. The cells were literally filling up with yellow trash bags. They were collecting garbage just fine, but they were completely failing to actually get rid of it. Now, this discovery was huge because it pointed towards a possible fix. I mean, if the problem is a failing cleanup crew, maybe you could give them a little boost. The researchers tried exactly that, using a drug called G2115, which is known to give this autophagy process a kickstart. Could this drug help the old cells finally take out the trash? And what was blocking the whole system to begin with? And this, right here, is the big aha moment of the entire study. The reason the trash wasn't getting processed, the reason the power plants were so stressed out, it wasn't a problem with the trash bags or the cleanup crew being lazy. It was something way more fundamental.
You see, the problem wasn't the trash bags at all. It was the recycling centers themselves. These recycling centers are called lysosomes, and they have all the acid needed to break down the waste. For the system to work, the trash bag and the recycling center have to meet.
And this is the fatal flaw in an old neuron. In young cells, the recycling centers are spread out everywhere, all along the neuron's long branches, right where the trash is being made. But in the old neurons, they've completely vanished from those branches. They're all stuck, trapped way back in the main cell body. It's like collecting trash all over a sprawling city, but the only recycling plant is stuck in one building downtown, and all the roads are closed. This was the bottleneck. This was the whole problem.
Okay, so now all the puzzle pieces start to click into place. If the real issue is this logistical nightmare, this waste management bottleneck, what happens if you can just power through it? This is where that G2115 drug comes back into the picture, and we get to see how fixing the cleanup crew can actually save those overworked power plants.
The results were honestly amazing. By ramping up the cleanup crew, the drug helped the cell overcome that recycling bottleneck. It specifically boosted the recycling of the damaged mitochondria. As those old broken power plants were finally cleared away, the ones that were left got healthier. Their glow literally shifted from that sick green back to a healthy red. And with that stress gone, they could finally relax. They no longer had to frantically split and merge just to stay alive.
And this chart right here is the smoking gun. On the left, you see that insane, hyperactive state of the mitochondria in the old neurons. But on the right, after a little help from the drug, that frantic activity drops right back down to the same calm level you see in a young, healthy cell. This was the proof. The mitochondrial stress isn't the cause of aging, it's a symptom of a broken recycling system.
So, after following all the clues, we can finally see the whole picture. This investigation gives us a totally new story, a new model for what's really happening inside our brain cells as they get older. We can basically rewrite the textbook.
In a young neuron, it's like a well-oiled machine. The recycling centers are exactly where they need to be. Waste gets taken out efficiently. Damaged mitochondria are recycled without a problem. And the power plants can just hum along at a normal, healthy pace. It's a system in perfect balance.
But in an old neuron, one simple defect sets off a disastrous chain reaction. The recycling centers get stuck. That creates a traffic jam for the whole waste removal system. Damaged power plants start to pile up like junk. And to survive in that toxic environment, the healthy mitochondria have to kick into that hyperactive compensatory state. The key takeaway here is that their hyperactivity isn't the disease, it's a desperate and frankly brilliant survival strategy.
And that brings us to the big conclusion. This isn't just some interesting story about cells in a dish. It's a fundamental rewriting of how we think about neuronal aging. It shifts the blame away from failing power plants and onto a simple logistical problem with waste management. And that is so exciting because it opens up brand new ideas for treating diseases like Alzheimer's and Parkinson's. But most importantly, it suggests that by fixing that one core failure, we might be able to actually reverse the damage. That reset button might not be science fiction. So the real question now is, what chapter comes next?